Awesome. So the purpose of this defense is for me to tell you all what I and my advisors and collaborators have discovered during the last 4 years of my PhD. After the presentation, we'll have a little bit of time for questions from the audience, and then I'm going to have to ask all of you to leave so my thesis committee can ask me the really difficult questions and decide whether or not I deserve a PhD in neuroscience. So before we get started, I just want to make sure everybody has their microphones muted so we don't have any accidental interruptions. If you're new to Zoom, that mute button is on the bottom left corner of the screen. It should have a red line through it if you're muted. All right. So we're going to start out with some background so I can explain to all of you what plasticity is and how we measure it in the human brain. And then we're going to talk about how exactly we track plasticity in our participants. And the bulk of the talk will be covering some of the results from this experiment and what we think these results tell us about how the brain functions. All right, so for some background, the first thing you need to know is that plasticity is a set of biological processes that reshape brain circuits and change the way they function. So plasticity is very important in all of our brains. It allows for brain development, allows us to acquire new skills and knowledge throughout our lives, and it can enable us to recover from even serious brain injuries. We induce plasticity in our participants by constraining their dominant arm for 2 weeks. The point of this cast was to stop them from using their arms, and therefore stop them from using the brain circuits that control their arms. And we thought that might induce plasticity in their brains. So since we're talking about motor plasticity, I should also teach you a little bit about motor anatomy. So we'll start out easy. This is a brain, and this is a spinal cord. This is an arm, all right? And our brains can control our arms by activating neurons in a part of the brain called the primary motor cortex. When these neurons are activated, they send an electrical signal down into the spinal cord, which is then carried on to our muscles and allows for our movements. Now, the motor cortex doesn't just control the arm. It actually has neurons that control almost every part of our bodies. And if you look at a slice through the motor cortex, all of those neurons are organized in a very specific way, such that neurons in this region of the brain will activate muscles in our face. Neurons over in this part of the brain will activate muscles in our legs. And this anatomy is very important for this thesis because we casted one upper extremity. And so we expected this, we actually knew that this would drive plasticity in the upper extremity region of the motor cortex. All right, the reason that we knew that it would drive plasticity there is because a similar experiment was done about a decade ago in squirrel monkeys. So just like us, they casted the arms of these monkeys for an extended period of time, and they tracked changes in brain function during casting. So the way they did this is different than how we will have done it. But what they did is they took a small electrode and they moved it along the surface of the upper extremity region of the motor cortex. And in each of those spots, they stimulated the motor cortex and then looked at what part of the animal moved. And there were lots of areas that shown here in red that caused finger movements. And then there are lots of other areas shown here in green that cause movements of other joints like the wrist or elbow. And the really interesting result from this experiment was that these areas that control the fingers, this finger representation, shrunk in the motor cortex after casting. This is called map plasticity, and it's one of the best studied forms of plasticity in neuroscience. And then after the cast was removed, that finger representation expanded again, and then the somatotopic map in that area returned to normal. All right, so experiments like this are great because they give us really detailed information about the cellular processes that underlie disuse-driven plasticity. But they have two big limitations. One is that it's really, really difficult to do experiments like this in a human. Most humans don't want you sticking electrodes into their brains. And the humans who do have electrodes put in the motor cortex are usually undergoing neurosurgery, and they're going to need intensive neurorehabilitation. And so they're not great candidates for an experiment that's explicitly designed to worsen their motor function. Another limitation is that this type of study focuses on a single brain region, in this case, the primary motor cortex. And the problem with that is the primary motor cortex doesn't function in isolation. In order for it to control our movements, it has to cooperate with the primary somatosensory cortex, the supplementary motor area, the cerebellum, thalamus, and basal ganglia. Together, all of these regions have to cooperate to coordinate our movements. And together we can refer to all of these regions as the upper extremity division of the somatomotor network. And the upper extremity somatomotor network doesn't function in isolation either. It has to cooperate with other subdivisions of the somatomotor network that control other parts of our bodies in order to coordinate complex movements. And all of these different divisions of the somatomotor network must cooperate with other sections of the brain, like the premotor network and the cingulo-opercular control network, in order to select actions that support goal-driven behavior. And to some extent, every part of the brain is connected to and interacts with every other part of the brain. And so each of these colors represents a different functional network, each with their own specific cognitive functions. We don't have time to talk about all those networks right now, but the point is that studies that focus on a single part of the brain are unable to describe the whole picture of plasticity. In order to understand everything that's going on in disuse-driven plasticity, we also need to understand how it affects the interactions between brain regions. And this is something that we can study very well in humans. So there are two methods I want to talk about for how we study brain activity in humans. One is called task-based functional MRI. So in this method, we ask people to perform some sort of behavior or cognitive task. In this case, someone's being asked to just move their right hand. And then we use MRI to track local changes in blood flow during their task performance. So in order for this person to move their hand, they have to activate neurons in the upper extremity somatomotor network. And that neural activity recruits more blood flow to the parts of the brain that support this behavior. And so if we look at the contralateral motor cortex, the left motor cortex, we see that there's a gradual increase in blood flow, followed by a brief dip in blood flow. And this is the very classic response that a brain region shows. This is a very classic change in blood flow we see anytime there's a brief burst of neural activity. But MRI doesn't just look at one part of the brain. One of the big advantages of it is that it shows us every part of the brain all at once. So we don't just see that the somatomotor cortex has this, this hemodynamic response function. We get to see that many other parts of the brain do too. And so this will show us that parts of the insula and the supplementary motor area are also involved in controlling this movement. So task fMRI is great in that it tells us a lot about the parts of the brain that are activated by a task. But in order to study every part of the brain, we would either need lots of tasks or to use a different modality. And so another way to study the organization of the brain is known as resting state functional MRI. So in this modality, we ask people to just lie quietly in the scanner. And we just record what their brains do at rest. And in all of our brains, all the time, every part of our brain is constantly active. So if we look at, say, the motor cortex here, and we just record from that area over time, we see all of these small fluctuations in blood flow. And those fluctuations in blood flow correspond to gradual changes in synaptic activity. The reason that resting state fMRI can tell us about the organization of the brain is because these fluctuations in activity are synchronized across functional networks. So if we look at another region of the somatomotor network, in this case, the opposite hemisphere motor cortex, we see that it also shows fluctuations in activity, and those fluctuations are highly synchronized with the left motor cortex. And we can quantify this synchrony as a Pearson correlation coefficient, and in this case, it's 0.7. So resting state fMRI can, just by having a participant lie quietly in the scanner, tell us about how different parts of the brain interact with one another and give us some sense of the whole of the organization of someone's entire brain just by asking them to sit still. And it's a little mysterious still, even though this is a method that's been widely used now for decades. It's still mysterious why functional connectivity even exists. What is all of this spontaneous activity? Why is it synchronized across brain networks? And so one prominent hypothesis to answer that question is to say that brain regions within a functional network are functionally connected because they are frequently co-activated during behaviors. So this can be called the Hebbian hypothesis of functional connectivity. And so to give an example, we use our left and right hands together all the time during our daily behaviors. And so this hypothesis says that this repeated co-use of our left and right hands co-activates our left and right motor cortex, and that repeated co-activation strengthens connections between the left and right motor cortex and produces strong functional connectivity at rest. Now, this is a hypothesis that is often stated in my field, but I would argue that until recently, there wasn't a lot of strong support for it. And this is one of the things that we wanted to change with our experiment. All right, so now for the fun part. We took 3 right-handed participants and casted their right arm for 2 weeks. The first participant was Nico, my PhD advisor, who all of you just got to hear from. Nico actually piloted this study on himself in order to show that it was safe, and that was why we were allowed to do this study in the first place. Second subject was a friend of mine, a graduate student with Brad Schlager at the time, named Ashley Nielsen. She's now a postdoc at Northwestern. But she was our second participant. And then our third participant wishes to remain anonymous, but we call him Omar after my favorite character from The Wire. Okay, so all these participants wore accelerometers on both of their wrists throughout the experiment. And these allowed us to track their daily behavior. Here I'm plotting a ratio of how many times each day subjects use their right hand to the number of times each day they use their left hand. So at baseline, this ratio is around 1 in everybody, meaning that they use their right and left hands about equally. But then during the cast period, this ratio goes down dramatically, indicating that the casts prevent them from using their casted arm. And that prolonged non-use or disuse led to functional degradation. So everybody lost a lot of grip strength. So here I'm plotting grip strength measured in both hands before and after casting. And you can see that in the casted arm, people lost about 40 pounds of grip strength. And unfortunately, that recovered fairly quickly afterwards. I should note, there are previous studies on immobilization that show that at least at 2 weeks, this kind of loss of strength is not primarily driven by muscle atrophy or changes in peripheral nerves, suggesting that this kind of change in motor function is likely due to plasticity in the central nervous system. So we want to study that plasticity. And so we acquired a lot of MRI data in this experiment. So this is an example schedule for the data we collected from Ashley. And this included 30 minutes of resting state fMRI every day for 64 days. Task fMRI before and after the cast. Diffusion tensor imaging, which allows us to look at white matter tracks that connect brain regions. I won't be talking about those data today. And periodic strength and fine motor testing before and after casting. And then, as I mentioned, constant behavioral monitoring using accelerometers worn on both wrists. Now, I should emphasize here that this is a very uncommon amount of data to collect in a human imaging experiment. Resting state functional connectivity is a noisy measurement, and the usual approach to overcoming this noise is to collect a little bit of data, like 5 minutes of resting state data, on lots of different people, and then average all of their brains together to get to reduce the overall noise. But the problem with doing this is that everybody's brain is a little different. And so when you average everyone's brains together, you get a sort of blurry mess that doesn't really look like any of the brains that went into the average. So this is true of a structural scan, like I'm showing here, but it's also true of these functional network maps. So here I'm showing the network maps for our 3 different participants. And you can see that there are some commonalities between these maps, and those are captured in this group average map. But there are also lots of unique features to each individual. And so our lab has spent a lot of time over the last several years pushing the message that it's important to study individuals separately. Because if you take this classic group average approach and you're interested in like this brain region right here, you will be studying different brain systems in different subjects if you just look at the same spot in every brain. And that's going to cause a huge source of unexplained variance in your data and could overwhelm whatever result you're actually looking for. The trade-off is that to study an individual like this requires a lot of data in that individual. So this is a study our research group did a few years ago that showed that to get a reliable estimate of functional connectivity in one person, you need between 30 and 100 minutes at least of data to make that estimate. So this is why we collected so much data in our participants before, during, and after casting. So now I want to show you what we can do when we have so much data combined with a powerful plasticity manipulation. Here I'm plotting out functional connectivity that we measured between the left and right motor cortex every day before casting. And so you can see that with 30 minutes of data each day, we get a very reliable measurement showing functional connection between these regions of around 0.7. And I said that going into this, we were hoping to test this hypothesis that functional connections are strengthened by co-activation during behavior. And so we had a prediction that blocking use of one upper extremity would block co-activation of this left and right motor cortex and lead to a gradual reduction in functional connectivity between these structures. Now, no one had really specified exactly what time course they expected this type of change to take, but we had always sort of assumed that these patterns of co-activation would be accumulated over a very long period of time, maybe over a person's entire life. So we thought 2 weeks of casting might be, if we were lucky, just enough to drive a measurable change in connectivity. And it was still possible that 2 weeks of casting wouldn't be enough to drive any effect at all. But that's not really what we found. This data point was collected after just 12 hours of casting. This was 1 day later. And then over the next 2 weeks, connectivity between the left and right motor cortex continued to decline. And then after we took the cast off, it returned to baseline very quickly. You can see that we have 1 data point after removing the cast where functional connectivity is still low. And then after that, we seem to be back up at baseline. So those data are just from 1 participant, but we saw very similar effects in all 3 of our participants. In all 3 cases, we drove a much larger effect size than we were expecting. Even in Nico, who had the smallest effect that we saw of a change in correlation coefficient about 0.23, this was still about twice the effect size shown in any previous study that attempted to modify functional connectivity using behavior. So just to make sure that this effect was genuinely specific to the upper extremity system that we had impacted and wasn't some sort of diffuse loss in connectivity because of something going wrong in their brains, we also tested connectivity between these neighboring regions of the motor cortex that control the legs. And as we expected, this didn't show any effect. So this showed that this effect was focal. All right, so next we wanted to really take advantage of the whole brain coverage of fMRI and examine disuse-driven plasticity beyond the somatomotor system. So to do that, we started by examining changes in functional connectivity between the disused motor cortex and the rest of the brain. So here, this blue dot shows the left motor cortex. This is the spot that controls the arm that will be casted. And the red and yellow areas in this map are regions that are strongly functionally connected with that left motor cortex. So you can see at baseline, the left motor cortex is connected to the rest of the somatomotor cortex, especially the corresponding region of the opposite hemisphere. This is pretty typical. Then during casting, we see an entirely distinct pattern of connectivity, where instead of being casted to the rest of the connected to the rest of the somatomotor cortex, this left motor cortex is now connected to areas like the supplementary motor area, the insula, the post-central sulci. And then after casting, we again see that sort of classic pattern of connectivity to the rest of the somatomotor cortex. So this map shows the changes in connectivity in all brain regions with the left motor cortex. So, you can see in blue that the rest of the somatomotor cortex decreased its connectivity with the disused motor cortex. And there are also many regions here in red that showed increases in connectivity. And to understand what all these regions are, we need to compare this map of whole brain changes in connectivity to this participant's individual specific network map. So this is what that looks like. On the left, I'm showing this network map in anatomical space. So this is this subject's cortical surface, and it's divided into many different brain regions. And the brain regions are colored depending on what functional network they're a part of. On the right, I'm also displaying all of those brain regions. Now each one is represented by a dot. And those dots are positioned using a technique called spring embedding. This technique treats functional connections between brain regions as springs and draws together closely connected regions. So that's why we see these clusters that correspond to the different known functional networks. So when we examine the changes in connectivity in this network space, the spring embedded space, we see that they are very focal in this space. So we see decreases in these 3 different clusters, which correspond to the 3 subdivisions of the somatomotor network. And then we see increases in essentially 1 cluster. Even though these increases are scattered around the brain in anatomical space, there's really only 1 region in network space that shows this increase in connectivity. And that region in anatomical space or in network space corresponds to this network known as the cingulo-opercular network. We can quantify the specificity for the cingulo-opercular network by examining the top 5% of brain regions showing increased connectivity with the disused motor cortex. And in all participants, those brain regions fall overwhelmingly in this one network, the cingulo-opercular network. All right, so far I've shown 2 surprisingly clean results involving changes in functional connectivity. But these are a little bit abstract. You know, we have some regions of the brain that are becoming more synchronized with one another, and some regions of the brain that are becoming less synchronized with one another. And it's a little difficult to wrap your head around what all of that means. And so we wanted to find a more concrete description of what was changing in our participants' brains. And so to do that, we went back to the original recordings we made from the affected brain regions. And this is what those look like. So each line here represents one 30-minute recording from the left and right motor cortex. The left motor cortex, the side that's affected by the cast, shown in black, and the right motor cortex is shown in red. And this is fairly zoomed out, so you can see lots of sessions. But I think that you can see that there's something abnormal happening in these sessions here, which are the sessions acquired during the casting period. And if I zoom in, you can see that that abnormal thing that's happening are these large pulses of spontaneous activity in the disused motor cortex. We can detect these pulses using a threshold-based method, and if I superimpose all of them onto one another, you can see that they have a very stereotypical shape, and that matches that hemodynamic response function that I was talking about before. This suggests that each of these pulses is a brief burst of neural activity in the disused motor cortex. And it turns out that these pulses don't just happen in the primary somatomotor cortex. This map here is a statistical map known as an analysis of variance, or an ANOVA, showing different regions of the brain that show consistent time courses surrounding each of these pulses. So this is showing that we also see pulses in the left somatomotor, or sorry, the left supplementary motor area, as well as the right cerebellum and the insula. So it's really the entire disused motor system shows these pulses. And they don't happen simultaneously in each of these regions. They actually start earliest in the supplementary motor area, and then they spread through the circuit. They go to the primary somatomotor cortex next, and then the cerebellum last. And these patterns were very consistent across our participants, both in the anatomical regions involved and in the actual magnitudes of the time delays between these regions. So in every case, you can see that we get these pulses starting in the left supplementary motor area, then spreading to the left somatomotor cortex, and then ending up in the right cerebellum. All right, so now let's talk about what all this means. So our first big conclusion from this study is that functional connectivity is highly related to a person's most recent behaviors and experiences. This was a surprise for us because, as I said, we were expecting that behavior had an impact on functional connectivity, but we thought that impact was averaged over very long periods of time. And so this is really important for interpreting studies that, say, scan patient populations, because it's possible that these differences in functional connectivity we see in patients might reflect disease-specific differences in behavior prior to scanning. So that could be an important clue to what those findings mean. Our next conclusion from this was that whole brain plasticity can be highly focal in network space. This didn't have to be the case. I've seen lots of studies that report changes in functional connectivity that are sort of scattered around the brain. They don't seem to correspond to the functional network architecture at all. And it makes you a little uncertain about what those effects mean and, you know, if they're real or noise. And so the fact that we see these changes so closely respecting these subjects' predefined functional networks really makes us feel more more secure that these are real findings. And it also sort of validates our network mapping approaches as showing something legitimate about the structure of the brain. It's also very interesting that the cingulo-opercular network in particular was involved here. This is a higher order control network that sort of exerts abstract control over all of our behaviors. And so we definitely wouldn't have known going into this that we would induce plasticity of this network by just casting one extremity. So understanding more about what is changing in this cingulo-opercular network during casting will be very important for creating new advances for neurorehabilitation. All right, and to me, the most exciting finding was of this novel form of spontaneous activity, these spontaneous activity pulses that propagated through the disused motor circuit. I'll admit that at first we didn't know exactly what these were, but we've been thinking about this for a long time, and so I want to share what we're thinking currently. The unique thing about these pulses is that they're sort of transient in their nature. Normally when we record spontaneous activity in the adult brain, it's made up of lots of little fluctuations that all combine together into this sort of stationary process. And these pulses are something that happen every once in a while, and then other times they're not happening. And so that's different. But it turns out that that is actually the norm for activity in early development. So if you look at the visual circuits in an animal very early in development, you'll see that they're usually silent. And then every once in a while, a big wave of activity comes through that circuit and depolarizes a large portion of the neurons in the circuit. Those neurons are then unable to fire for a period of time, and that leads to these periods of silence in between waves of activity. And in the visual system, these waves are called retinal waves, and we know that they're very important for normal visual development. If you block the generation of these waves early in the development of a mouse, you end up with an adult mouse that has strong visual deficits. And even though these waves are best studied in the visual system, we know that they also happen in other parts of the brain as well, including the somatomotor system, the auditory system, and virtually every other part of the brain. And so as development progresses, these waves become more frequent and smaller. And so spontaneous activity goes from having this sort of transient every once in a while to this more stationary oscillation that we see in adults. And part of the reason for this change in the dynamics of spontaneous activity is because of use-dependent maturation of inhibitory interneurons. As cortical inhibition matures, it tamps down these waves and stops them from depolarizing large portions of the neural population. And that prevents those refractory periods that create the transient dynamics we see in early development. But if an adult circuit undergoes a period of disuse, previous studies show us this leads to a focal reduction in inhibitory tone in those disused circuits. And I would like to suggest that this focal disinhibition might support the emergence of these spontaneous pulses of activity in the adult brain. We know that these waves of activity in early development are important for refining neural circuits and allowing them to develop normally. And if these disuse-driven pulses that we see in the adult brain are similar to these waves of activity, which I'll admit is still a big if, then it may be the case that these pulses can help maintain these circuits in the absence of regular use. Our lab is very excited about this possibility, and we're doing a lot of work to follow up on it. There is a lot left to do to test this potential relationship between these adult pulses and these developmental waves. But this line of thinking has already led us to one prediction that we've been able to test with our existing data. So the really key feature of this developmental spontaneous activity that allows it to refine these circuits is that it propagates as a wave across brain regions. So here's a retinal wave on the left. This is a time course of the retinal wave. So each of these videos, each of these photos is like a form of video. Sorry. And you can see that in both the primary visual cortex and the superior colliculus, another part of the visual system, these waves emerge in one part of the region, and then they propagate across the region. And this happens simultaneously in multiple parts of the system. There's a lot of computational work that shows how this sort of wave-like propagation across brain regions can help refine the organization of circuits. And there's also experimental evidence to show that if you disrupt this wave-like propagation, you don't get normal visual development. And so if we think that spontaneous activity pulses in the adult brain can help maintain circuits the way that these developmental waves of activity can refine them, then those spontaneous activity pulses would also need to show this wave-like propagation within brain regions. So here's a map that shows how early or late pulses occur in every part of the brain. And what you can see here is that within each region of the brain, there's a wide range of timing of these pulses, such that in every case, they happen early in one part of the region and then move across that region. So we think that this wave-like propagation could help maintain these circuits. It can be a little difficult to picture these waves based on maps like this. And so we've come up with a more intuitive way of visualizing this. And so this is what these waves look like. They start in each of these brain regions and they sweep across the region. And they do this in multiple regions in synchrony. Again, we think that this could support maintenance of these circuits in the absence of regular use. If that turns out to be true, then we may have uncovered a mechanism by which the brain can maintain its own circuits in a sort of standby mode when they're not being used. There's a lot of work left to do with this. We are going to continue working on this, and we hope that others will as well. But we think that there's potential here to uncover something that could really be transformative for neurology and neurorehabilitation.